Hello, and we are back with the second part of this mind map series. So, let's have a quick restart for this by taking our highlighter and reading the things and understanding them in a proper way. Cell wall. It is a non-living rigid structure called the cell wall, an outer covering of the plasma membrane in fungi and plants. The cell wall is young plant cell. The primary wall is capable of growth, which is gradually diminished as the cell matures, and the secondary wall is formed on inner side towards membrane of the cell. The middle lamella is made up of calcium pectate. Algae have a cell wall made up of cellulose, galactons, mannans, and calcium carbonate. In plants, it consists of cellulose, hemicellulose, pectin, and proteins. Endomembrane system. Each of the membranous organelles is distinct in terms of the structure and functions. Many of these are considered together as an endomembrane system because their functions are coordinated. We have certain things such as endoplasmic reticulum, Golgi apparatus, lysosomes, and vacuoles. We'll be reading their details line by line, starting from the endoplasmic reticulum. It is the reticulum of tiny tubular structure scattered in the cytoplasm is called ER or endoplasmic reticulum. The ER or the endoplasmic reticulum, which has ribosome on surface, are called RER, rough endoplasmic reticulum. In absence of ribosomes, they appear to be called as SER or smooth endoplasmic reticulum. RER is involved in the protein synthesis, whereas SER is involved in the lipid and steroid synthesis. These were named Golgi bodies after discoverer named Camillo Golgi. They consist of cisternae, which are centrically arranged near the nucleus with convex cis or the forming phase, and concave trans or the maturing phase near the plasma membrane. This says about the Golgi apparatus. Principally performs the function of packaging of materials, is important site for formation of glycoproteins and glycolipids. Further moving to the lysosomes. These are the membrane bound vesicular structure performed by the process of packaging in the Golgi apparatus. They are rich in hydrolytic enzymes, lipase, protease, carbohydrates, optimally active at acidic pH, that is, the pH is less than 7. As we know, the 7 is the neutral pH. These enzymes are capable of digesting carbohydrates, proteins, lipids, and nucleic acids. Further talking about the vacuole. Vacuole is the membrane bound found in the cytoplasm. Membrane is coltonoplast. Contain water, sap, excretory products, and other materials not useful for the cell in amoeba. Contractile vacuole is more important for secretion. In many cells, as the protist foods, vacuoles are formed by engulfing of the food particles. It is seen that in some of the plants, vacuole acquires more than 90% of the concentration itself alone. Moving on to the mitochondria, also known as the powerhouse of the cell, as it causes the ATP synthesis. Mitochondria, unless specifically stained, are not easily visible under microscope. Each mitochondria is double membrane bound structure with inner compartment called matrix. The two membranes have their own species called enzymes. The outer membrane forms the continuous limiting boundary of the organelles as and internal membranes form the cristae. Mitochondria at the site of mitochondria are the sites of anaerobic respiration. They produce cellular energy in the form of ATP, hence called powerhouse of the cell. The matrix has single circular DNA, a few RNA molecules, ribosome 70S, and the components required for the synthesis of protein here. The Swedenborg's unit, stands for the sedimentation coefficient as it is the indirectly measured of density and size. Plastids. Plastids are found in all plant cells and is euglenoid based on the pigments. Plastid can be classified into chloroplast, chromoplast, leucoplasts. The chloroplast contains chlorophyll and the carotenoid pigments. The leucoplasts are colorless plastids, amyloplasts, and store carbohydrates. Example: potato. Aleoplast stores oil and fats, whereas the uroplast stores protein. Chloroplasts are also double membrane bound structure, which has the membrane sac-like structure called thylakoids that are arranged in such arrangement in a thala arrangement. Not thala, it's thala. And the matrix is called stroma. It also contains small double-stranded circular DNA and ribosome. Carotenoid is fat-soluble pigment called keratin and xanthophylls. The ribosome of the chloroplast 70S are smaller than cytoplasmic ribosome. Thylakoid are arranged in the stacks called grana, singular granum, flat membranous tubule called the stroma lamellae connecting the thylakoids of the different grana. Stroma require enzymes for carbohydrate and protein synthesis. Chlorophyll pigments are present in the thylakoids. Now only one page is left for the mind map series, and we'll be completing this by the next video. Till then, stay tuned and stay happy. This was the second part of this lecture, completed successfully, and one part more will be coming. Till then, thank you.